Namaskar. Today we will study cell cycle. So as we know, when a cell undergoes cell division, a parent cell gets divided into two daughter cells. So from one cell division to the next cell division, the stages which a cell undergoes are called as cell cycle. So what are the stages? Two stages or two phases. One is interphase and the other is M-phase or mitosis. Interphase is the period of preparation for cell division. First it prepares itself, then the actual cell division is the M-phase. And this interphase consists of three phases: G1 phase, S phase, G2 phase. G1 phase occurs for 8 to 10 hours. However, it can be in days or weeks or even years also, G1 phase. But mostly it is 8 to 10 hours, S phase is 6 to 8 hours, G2 phase is 4 to 6 hours. So S stands for synthesis of DNA. Before S phase, there is pre-DNA synthesis where there will be mainly organelle duplication. In the S phase, there will be duplication of the chromosome, and in the G2 phase or post-DNA synthesis or preparation for mitosis, there will be mainly cytoplasmic growth. So after the interphase, the cell is prepared and the cell undergoes mitosis, and in the mitosis there are four phases: prophase, metaphase, anaphase, telophase, and then from one parent cell we can have two daughter cells. So the cell cycle, you can show it in the cyclic form: G1 phase, S phase, G2 phase, M phase, and after the M phase the cell gets divided and two daughter cells are produced, and those two daughter cells then will undergo G0 phase or resting phase. G0 phase means the cells are not dividing but can re-enter the cycle into the G1 phase, and for some cells like nerve cells, skeletal muscle fibers, cardiac muscle fibers, they are permanently arrested in this G0 phase. So there will be those cells, those permanent cells, they do not undergo cell division. So once they lost, means they are permanently lost. Then the M phase consists of prophase, metaphase, anaphase, and telophase. So now we can see that here some positive regulators of cell cycles are there, like cyclin and cyclin-dependent kinases. They are called the positive regulators of cell cycle. They move the cell from the G0 phase towards the G1 phase, followed by S phase and G2 phase, and finally M phase. We can see here there are two checkpoints. One checkpoint 1 is before the S phase and checkpoint 2 is before the mitosis. And the role of these two checkpoints is to check whether a normal cell is going to be formed or abnormal cell is going to be formed. If abnormality is there, abnormal cell is going to be formed, then it will undergo apoptosis by the negative regulators of cell cycle. These negative regulators of cell cycle, they stop the cell division at these checkpoints and the cell will undergo apoptosis. What are the negative regulators? P53 gene, retinoblastoma RB gene are the negative regulators. In addition, we can have inhibitors of CDK, inhibitors of kinases, CDK inhibitors, and P21, P27 are the examples of some of the CDK inhibitors. And how this occurs, the cyclins, actually the cyclin-dependent kinases or CDK are inactive, and they become active when they bind to the cyclin. They form complex with cyclin, and the cyclin CDK complex is active, and there are different cyclins like cyclin A, cyclin D, cyclin E, cyclin B. Similarly, there are different CDK, CDK1, CDK2, CDK4, CDK6, and they form complexes, and those complexes regulate cyclin D, CDK4 or 6, cyclin E, CDK2 regulate transition from G1 to S phase, cyclin A, CDK2 responsible for your DNA synthesis, they regulate the S phase, cyclin B, CDK1 complex regulate progression from G2 to M phase, and some of the CIP or CDK inhibitor protein or CDK inhibitors like P27, P16, P21, they inhibit these complexes. P27 inhibits cyclin A CDK2, P27 inhibits cyclin A CDK1 complex, P16 inhibits cyclin D CDK4 complex, and cyclin D CDK6 complex. P21 inhibits cyclin E CDK2 complex. P21 also inhibits cyclin B CDK1 complex, and in this way they can stop the progression of cell cycle from G1 then to S to G2 and then finally M. And the negative regulators like P53 and retinoblastoma gene, they also inhibit the cell division, and mostly the retinoblastoma protein has a role in S phase gene transcription, and actually the retinoblastoma protein inhibits the regulatory transcription factor E2F, and so this retinoblastoma protein is inactivated by phosphorylating this protein. And who inactivates this? G1 cyclin CDK complex or cyclin D CDK4 complex, so that the retinoblastoma becomes inactive, and this regulatory transcription factor E2F can cause S phase gene transcription. So RB gene acts as a negative regulator. Similarly, the P53 gene P53 gene is usually complexed with MDM2, and when this P53 is active, that means when this MDM2 is released, and this can be released by two means: when there is excessive MYC, and when there is DNA damage, and this P53 actually regulates the transcription of P21, one of the CDK inhibitor protein, which will inhibit the cyclin E and CDK2 complex, as well as cyclin B and CDK1 complex, and will arrest the cell cycle in either G1 phase or G2 phase, and hence acts as a negative regulator of cell cycle. So this is all about cell cycle. Thank you.